Hello, and welcome to your Microbiology Bio 203 video lecture on biotechnology and DNA. I'm Mr. Kennedy, and I'll be your guide as we explore this issue together. Biotechnology is the use of microorganisms, cells, or cell components to make a product. Foods, antibiotics, vitamins, and enzymes are just a few things that can be made using biotechnology. Recombinant DNA, or rDNA technology, involves the insertion or modification of genes to produce desired traits. Often, a vector will be needed when inserting genes of our own choosing into a new host. A vector is a self-replicating DNA molecule used to transport foreign DNA into a cell. Clone is a population of genetically identical cells arising from one cell. Each clone typically carries the vector. This is an illustration of a typical genetic modification procedure. Tools of biotechnology. To understand the tools of biotechnology, we must first dive into selection and mutation. Selection is the selecting of a naturally occurring microbe that produces a desired trait. Mutation. Mutagens can cause mutations that might result in a microbe with a desirable trait. Site-directed mutagenesis is a targeted and specific change in a gene. Restriction enzymes are used to cut specific sequences of DNA. Restriction enzymes destroy bacteriophage DNA in bacterial cells. They function as a sort of an immune system for a microbe. Methylated cytosines in bacteria protect their own DNA from digestion. Restriction enzymes can create blunt ends or staggered cuts known as sticky ends. This is an illustration of 4 commonly used restriction enzymes and the sequences that they produce their cuts at. This is an illustration of a restriction enzyme's role in making rDNA. Here you can see the restriction enzyme is used to cut a piece of DNA away from a larger strand. The cuts produced a DNA fragment with sticky ends. The same restriction enzyme would be used in the extraction of DNA from another source, so that the two types of DNA will have complementary sticky ends. When the two fragments, both treated with the same restriction enzyme, are introduced to each other, the complementary sticky ends seek each other out. The joined fragments will usually form either a linear molecule or a circular one. Vectors carry new DNA to desired cells. They must be able to self-replicate. Plasmids and viruses can be used as vectors. Shuttle vectors exist in several different species and can move cloned sequences among various organisms. PCR, also known as polymerase chain reaction. PCR is the process of increasing small quantities of DNA for analysis. It's used for diagnostic tests for genetic diseases and detecting pathogens. Reverse transcription PCR uses mRNA as a template. This is an illustration of the typical PCR reaction. Genetic modification. In genetic modification, we can insert foreign DNA into cells. DNA can be inserted into a cell by transformation, electroporation, or protoplast fusion. Transformation is accomplished when cells take up DNA from the surrounding environment. Electroporation is accomplished when electrical current forms pores in a cell membrane and DNA is introduced through those pores. Protoplast fusion involves removing the cell walls from two bacteria, which allows them to fuse to each other. This is an illustration of protoplast fusion. Inserting foreign DNA into cells can also be done using a gene gun or microinjection. Genomic Libraries. Genomic libraries are collections of clones containing different DNA fragments. An organism's DNA is digested and spliced into a plasmid or phage vector and introduced into bacteria. At least one clone exists for every gene in the organism in a genomic library. Complementary DNA, or cDNA, is made from mRNA by reverse transcriptase. Complementary DNA is used for obtaining eukaryotic genes because eukaryotic DNA has introns that do not code for protein. mRNA has the introns removed, coding only for the protein product. This is an illustration of how cDNA, or complementary DNA, is produced for a eukaryotic gene. Synthetic DNA. Synthetic DNA is produced as genes using a DNA synthesis machine are constructed. This is an illustration of a DNA synthesis machine. The operator can program in and synthesize DNA fragments of their own choosing. Selecting a clone. Blue-white screening. Blue-white screening is one method of selecting clones. Blue-white screening uses plasmid vectors containing ampicillin resistance genes and beta-galactosidase genes. Bacteria is grown in a medium containing ampicillin and X-galactidase, a substrate for beta-galactidase. This is an illustration of blue-white screening, one method for selecting recombinant bacteria. Colony hybridization. Colony hybridization uses DNA probes, which are short segments of single-stranded DNA complementary to a desired gene. This is an illustration of colony hybridization using a DNA probe to identify a cloned gene of interest. Applications of DNA technology. Therapeutic applications of DNA technology include the production of human enzymes and other proteins such as insulin. Subunit vaccines are also an example of a therapeutic application of DNA technology. Subunit vaccines are made from pathogen proteins in genetically modified yeasts. Non-pathogenic viruses carry genes for pathogens' antigens as DNA vaccines. And gene therapy. Gene therapy is used to replace defective or missing genes. This table is an illustration of some pharmaceutical products that are produced using recombinant DNA. Other therapeutic applications of biotechnology and recombinant DNA include gene silencing. In gene silencing, small interfering RNAs, or siRNAs, bind to mRNA, which is then destroyed by RNA-inducing silencing complex. RNA interference, or RNAi, inserts DNA encoding siRNA into a plasmid and transfers that into a cell. Genomic projects. A number of genomic projects have been carried out for the past several decades. Shotgun sequencing sequences small pieces of genomes which are assembled by a computer. Metagenomics is the study of genetic material directly from environmental samples. The Human Genome Project is probably the most famous genome project, which sequenced the entire human genome. The Human Proteome Project maps proteins expressed in human cells. Scientific applications of biotechnology include bioinformatics. Bioinformatics is the understanding of gene function via computer-assisted analysis. Proteomics is used to determine proteins expressed in a cell, and reverse genetics, which allows us to discover gene function from a genetic sequence. Southern blotting is commonly used in scientific applications of biotechnology. In southern blotting, DNA probes detect specific DNA fragments, known as RFLPs, and then are separated by gel electrophoresis. This is an illustration of the Southern blot procedure. Forensics microbiology is also another application of DNA technology. DNA fingerprinting is used to identify pathogens. PCR microarrays and DNA chips can screen samples for multiple pathogens. It differs from medicine because it requires proper collection of evidence and establishing a chain of custody. This is an illustration of a DNA fingerprint. Nanotechnology. Nanotechnology involves bacteria that can make molecule-sized particles. Nanospheres can be used in drug targeting and delivery. Finally, agriculture. The TI plasmid occurs in Agrobacterium. It integrates into the plant genome and causes a tumor-like growth. The TI plasmid can be used to introduce rDNA into a plant. This is an illustration of how the TI plasmid can be used as a vector for genetic modification in plants. Another agricultural application is Bt toxin. Bt stands for Bacillus thuringiensis. We can also introduce herbicide resistance and suppress genes. We can increase the nutrient value of agricultural plants and even insert human proteins. This is an illustration of some agriculturally important products of rDNA technology. And this concludes our coverage of microbial, DNA, and biotechnology.